So, this completely different topic of methodological interest related to bacterial vesicles. We think this is interesting because vesicles has a potential of like a tool of communication, and how can bacteria affect our bodies. So now we're happy to hear a story about this from Helene Haugsten from Institute of Oral Biology. Please.
Okay, thank you. Today I'm going to present some results from my ongoing PhD study. It's about periodontitis, and periodontitis is a chronic inflammatory disease affecting the supporting tissue of the teeth. And here in this figure you can see teeth and the bone, and at this side you see healthy gum and the healthy bone level. And on the right side here, you can see how it looks like when you have periodontitis. And then you can see this reduced bone level, and the disease contributes to the reduced bone level. Periodontitis is a polymicrobial disease, and it is related to systemic diseases, such as Alzheimer's and cardiovascular disease. And it is a common disease worldwide. Approximately 50% of the adult population in the Western world has some degree of the disease. And then we have P. gingivalis, a keystone pathogen in periodontitis, and central in the keystone pathogen hypothesis. And the hypothesis holds that P. gingivalis alone and in low abundance can lead the microbiota into a dysbiotic one. And P. gingivalis is also produce potent virulence factors. So in this study we aim to isolate the bacterial extracellular vesicles from 3 different strains of P. gingivalis. We wanted to characterize the vesicles and investigate the uptake of vesicles into human oral fibroblasts. And our objective is to better understand the role of P. gingivalis in the pathogenesis of periodontal disease. So all bacteria and cells release extracellular vesicles, and here's an overview of the main types of extracellular vesicles. Here you can see microvesicles budding from the plasma membrane. We have exosomes, who are believed to be of endocytic origin. We have apoptotic bodies from cells undergoing apoptosis. We have extracellular vesicles from parasites, and the bacterial extracellular vesicles, which is the main focus of my PhD project. So all bacteria, both pathogenic and non-pathogenic, release extracellular vesicles. And here you can see the membrane of a gram-negative bacteria, and you can see the bulging of the membrane forming an extracellular vesicle. And the way the bulging forms an extracellular vesicles contributes to the content and composition of the extracellular vesicles to be quite similar to the parent bacteria. And here at the right, you can see a nice scanning electron microscope picture of an bacteria, and you can see the extracellular vesicles budding on the outside of the bacteria. The vesicles are believed to have many important functions. Here is an overview. We have physiological functions such as cell communication and signaling here, and we have pathological functions such as delivery of virulence factors. And all these functions are possible due to that the vesicles can carry most of the same cargo as the parent bacteria. So here in the middle you can see an illustration of an extracellular vesicles from a bacteria and its cargo, like RNA, DNA, proteins, outer membrane proteins, and virulence factors. So this is a short overview of my project. I'm going to present results from 1 and 2 today. So first we culture the P. gingivalis, And then we want to isolate the bacterial extracellular vesicles, and we do that in 2 steps, ultrafiltration and site exclusion chromatography. And then we want to characterize the vesicles, and we do it with nanotracking analysis, transmission electron microscopy, and flow cytometry. And the second, we have done some studies on the uptake of the vesicles to fibroblasts. Okay, so first culturing of the vesicles. We have chosen 3 different bacterial strains from P. gingivalis with different virulence. And we grow them on these blood agar plates. But since this blood agar contains vesicles, we swap the bacteria into the brain-heart infusion medium after 5 days and grow them further anaerobically. And along the way, we determine the optical density to make sure that we harvest the vesicles in the log phase of the bacterial growth and before the stationary phase. And then we want to isolate the vesicles. We do that in 2 steps. First, we do the ultrafiltration, where we concentrate the supernatants through several steps. And then we do size exclusion chromatography, and the particle pass the columns based on their size, and that leads that means that the vesicles can be separated from the proteins, for example. So now we have isolated the vesicles, and then we want to characterize them. And the first thing we do, we use nanotracking analysis to measure the size and concentration of the vesicles. And in this graph here, you can see one sample of a bacteria, and the mean size of the vesicle is 110 nanometers. And we often found the vesicles to be from 50 to 200 nanometers. And then we do transmission electron microscopy. And we take our samples and do negative staining and take pictures. And here you can see 3 pictures from 3 different strains, over 3 different strains, and you can see vesicle-like structures here, and also here. And then we do flow cytometry, and we do the flow cytometry targeting gingipains. And gingipains is known wireless factor specific to P. gingivalis. So here you can see the fluorescence here and the event count here. And the control is the red peak, and the green peaks are the bacterial strains. And this shows that the fluorescence is lower in the control than the bacterial strains, and that means that we possibly have gingipains on the outer membrane, now on the bacterial vesicles. And the last results I'm going to show you is the uptake of extracellular vesicles in fibroblasts. So we labeled the vesicles with PKH67, the green color, and the nuclei with DAPI and the blue color, prior to imaging with confocal microscope. And here we can see possible uptake of the vesicles to the oral fibroblasts. And in the end, this is our next steps in our project. We are now focusing on the effects of the vesicle uptake on cell function. So nowadays we do proliferation, migration, and viability studies. We also want to look at the content of the vesicles. We have done proteomics, and we also hope to do metabolomics later. And thank you to all my supervisors and everybody at the lab. And I can take questions if you have any.
So thank you very much for a nice presentation about a different topic. Questions from the audience? I guess I'll keep asking questions then. So what do you think are in these vesicles? I know you'll investigate it later on, but...
They are believed to have much of the same content as the parent bacteria. So we have the proteins in the membrane, and the RNA and the DNA, and the virulence factors. That is very central.
So, but do you think these vesicles, they act, in general, will they act just locally, or do you think will they, you believe mostly that they will affect the human cells, or will they affect other bacteria, or do you think they can be taken up in blood, or what do you?
I think both, and it's there are several things we don't know about the vesicles and how they work, but we know that they can be taken up, and also that they can travel to distant sites.
Yeah. So, because it's of course of interest to know what this is. We heard about chemistry earlier today, but whether these vesicles can be driving disease somehow. Are all types of bacteria making these vesicles, or is some? Do you know, are there some more than others? Do you have any idea about this?
I think what we know is we believe that all all bacteria release vesicles, both pathogenic and non-pathogenic. So and we see that, it's been shown earlier that the vesicles may be more potent than the bacteria itself.
Right.
Maybe.
So how do you show that to?
I didn't show that,
No.
but some other did.
Yeah. So, but to find my final question, do you have a question, Beate?
Thank you for a very nice talk. So what would be your next step? So methodologically, what kind of analysis would you like to do to try to characterize the insides of these vesicles?
We have done proteomics and hopefully metabolomics later, but we have done proteomics, and it's a very, very early stage of the analyzing, but it looks like we have some of the same proteins as in peach valleys in the vesicles.
So are they more potent than the bacteria? Or?
We haven't studied that, no. But we'll also do viability and proliferation and migration and see if we have any results from that.
Thank you.
Phil?
Phil Pope from NMBU. I had a question regarding if you got any ideas on what triggers their production, because a lot of people assume they just kind of happen when the cells get old and age, but there's data from cellulose degrading bugs from herbivores which will show that they're actually induced by a substrate. So if you grow these bugs without a substrate, they won't produce vesicles, but if you add the substrate they want, then they'll start making them.
We know that this release of vesicles happens by themselves, but we also know that several things can affect the release, like temperature and stress can do like that we get more release of vesicles. If that was the answer.
So here you are looking at some specific bacterial strains. Do you plan to look at more like human samples to isolate from periodont? Is that feasible, sort of?
To take patient samples?
Take patient samples, isolate vesicles, and investigate those?
We don't have any plans now to do that, but that will be interesting. Yeah.
Okay, very good. If there are no more questions, and thank you very much.
Thank you.